Well, thank you. Thank you very much for the invitation. I'm very happy to tell you about this story and this paper that was recently published. This is done that it was work that was done during my postdoc at the Max Planck Institute in Munich, in Germany. And now I'm an assistant professor at the Karolinska Institute in Stockholm. So I'll tell you a little bit about the story. We'll go to keep the slides short, and so we can hopefully have a nice discussion. Okay, so let me see. Perfect. So I'm not going to tell you much about ketamine. I think everybody who is here knows about it exactly. Probably know it better than me, so I'll skip to this part. But I will focus on this. And although there's a lot of interest in ketamine and things are very they look very promising, there's still a lot of questions, including what is the issues with what is the best type of protocol to be administered to patients, concerns with side effects. But the one thing that we know right now is that an exact mechanism of antidepressant action is yet to be determined. Now, there's been a lot of work done on this topic of what are the actual mechanisms of ketamine, and a lot of beautiful work over the years. And so this is a little cartoon from one of the reviews for the Ronald Duman's lab. And here what we see is a lot of proposed mechanisms of action. Now, the hypothesis that we were driving this project with, it was possible that the more elusive molecular components of this mechanism could still be unidentified due to very important methodological limitations. And one of them is absence of cell type specific information. So most of the work that has been done in the field up until that point relied on either candidate gene studies or RNA studies when you looked at sequencing technologies at the bulk level, meaning that you can obtain information of thousands of genes, but looking at a homogeneous tissue of thousands of cells. So it's a mixed signal. And we thought that utilizing new developments in technology, such as single cell RNA sequencing that would give you cell type specificity, would give us a little extra information that we could use to try to understand what was happening downstream of treatment. Now, the project was very simple. In general, we basically took mice, we treat them with ketamine. We waited 2 days, and then we dissected the ventral hippocampus, and we prepared some single cell suspension. So a couple of things here to mention. Number one, the region. We focus on the ventral hippocampus. It's one of the regions that have shown to be involved in the mechanisms of action of ketamine, alongside with the cortex and a few other regions. But also the time. So this experiment is done 2 days after a single injection of ketamine, which speaks that we were interested in the sustained antidepressant effects of ketamine rather than the fast acting. And that's something that I will build, and we can talk about it later on. All right, so after doing this, we perform a single cell experiment. We obtain cell type specific information. If you're not familiar with single cell data, we're seeing clusters. Each color represents a different cell type, and we can obtain information, gene expression at the population level. We were able to identify different transcriptional signatures that were associated, again, with the sustained antidepressant effects of ketamine. And we found differences among different clusters. However, the largest difference that we found were among glutamatergic neurons. And because of this, we decided to try to validate these findings. And so to validate single cell data at that time, it was quite complicated. It's one thing when you're validating a specific pattern of cells that emerges, where you can identify globally. But when you're looking at relatively small changes across cell types, that could be quite different. You cannot do it by it's difficult to do it by immunostaining techniques or something like that. So we had to be a little bit creative. We developed these mouse lines where we can genetically label all glutamatergic neurons in the forebrain. That includes the hippocampus. We basically took this line, which are called NEXT-GRI-9, and we inject them with ketamine, same thing that we did our previous experiment. Now, the procedure is the same. The only difference is that instead of doing a single cell experiment, we take those cells and then we isolate neurons. Glutamatergic neurons were expressed in TD tomato and then everything else. So you will have cells expressed in TD tomato in one component and the remaining of the cells in the other component, meaning that it's not cell type specific, but it is at the population level. No, it's not single cell. And then we can validate that at the level of qPCR. So what we're able to find is we took 8 genes, random genes that were differentially expressed. So you picked 4 genes that were upregulated and 4 genes that were downregulated for the glutamatergic neurons to see if we could validate these findings. And indeed, if we look in the positive cells, we could see that there was not only a significant effect in some of these genes, but also the directionality of change also matched, where the upregulated genes were upregulated and the opposite for the downregulated. We also know that in the compartment of cells that were TD tomato negative, that did not include glutamatergic neurons, we did not see any of these changes. So we knew that these changes were cell-type specific as well. Now, one of the genes that was the strongest gene in our analysis was this gene known as Kcnq2. And I'll build a lot more because this is the beginning of this whole story. We basically focused on this gene then to try to validate it different ways. So one of the things that we did is we used an in vitro system to see what happens if you treat primary neurons from mouse hippocampi, and then we treat them with either ketamine or Huanqing, which is the metabolite. And what we can see is that after treatment with both ketamine or Huanqing, we could see an increase in the expression of Kcnq2 after 2 hours and even after 2 days later. So this was consistent what we were seeing in vivo, and suggested that Kcnq2 could be an important target for ketamine action. Now, what do we know about Kcnq2? Well, it's a potassium voltage-gated channel. We know that it plays a critical role in the regulation on neuronal excitability. In combination with Kcnq3, forms a Kcnq channel. And this channel forms a specific signal that is known as the M channel or the M current. And what's important about this is that we can isolate and we can manipulate this. And that was very useful for us. Now, this gene has been mostly associated with epilepsy, but there were, at the time when we started experiments, a few studies were emerging now implicating both Kcnq2 and Kcnq3 with stress in mice, as well as mechanisms of active resilience. So this is some of the work of Alison Friedman and James Moore in New York. Now, so as I said, we can isolate this M current, and so that's something that we wanted to see. So we took the same in vitro system. These are mouse hippocampi neurons. We treated them with ketamine. And what we can see is we saw an increase in the current density of Kcnq channels. Now, I should mention Kcnq channels, when we measure the M current, we cannot differentiate between Kcnq2 or Kcnq3. It's the signal of the channel. But we were seeing a difference. And we were able to replicate that in vivo. We took mice, treated them with ketamine, waited 2 days, basically the same protocol as we've done before. And then we do patch clamp experiments, we'll do ex vivo recordings, and then we could see same thing that we saw in vitro, which is an increase in the current density. Now, suggesting that the Kcnq2 mRNA expression that we observed in vivo, it's complemented by a significant gain in the number of Kcnq channels, both in vitro and ex vivo. Now, again, it supports the idea that ketamine regulates Kcnq2 expression, and that perhaps modulation of these channels in glutamatergic neurons could be a potential druggable target for MDD. So this is a good point where we were very happy to see consistent results both in vitro and in vivo, but we wanted to go a step further and then functionally manipulate these channels. So for this, we created some shRNA constructs where we tried to knock down the expression of Kcnq2. So we validated this in vivo and in vitro. And what you can see here is 2 constructs that we designed. We can knock down the expression about 50%. And this is important, that is not a knockout, it was a knockdown. If we were to knock out the expression of Kcnq2, this would lead to epileptic seizures in the mice. So that's definitely not something that we wanted. We need a functional Kcnq channel. So we did that, we knocked down the expression roughly 50%. And what we did is we took 2 groups. Some of the mice were treated with a control shRNA versus Kcnq2 knockdown. And then we randomly put some of these mice in a group treated with ketamine or saline, same as we've done in the past. The idea here was to assess antidepressant-like effects in mice. And for this, we use a forced swimming test. And what you see here is the mice who were treated with a control shRNA. And we see a decrease in the time that the animal spends immobile during the test. And this is usually associated with a response to antidepressant treatment. What's interesting is when we knock down Kcnq2, then we see that we could completely abolish these behaviors. Again, this is 2 days after treatment, same as we've done for the molecular changes. Now, this indicated that the ventral hippocampus is an important site for Kcnq function, and that this could be important also for the antidepressant effects of ketamine. All right, so up until now, I've shown you experiments in mice that were naive. These mice were not stressed, were not showing any depression-like phenotypes at all. And so what we wanted to do is what happens if we stress chronically mice, and if there leads to any changes in this potassium channel. And that's what we saw. We basically took the same mice that I mentioned earlier when we labeled glutamatergic neurons. This is an X3 line. We put them through a protocol called the chronic social defeat model that induces long-lasting depression-like phenotypes in mice. And what we see is once we put them through a forcing test, we see an increased effect, which is again is associated with a more depressed-like phenotype. But what's interesting here is that when we measure the expression of Kcnq2 in the positive TD2 meta-positive cells, we see a decrease. Now, just to remind you, this is exactly the opposite that we saw once we treated mice. We saw an increase of Kcnq2 and a decrease in the time in model. So this shows the opposite. What's also interesting, again, we did not see any changes in the TD2 meta negative cells. And when we measured, because this is done at the population, cell type specific again, when we look at the bulk level, meaning we just took the tissue, mixed tissue, and looked at Kcnq2 expression by qPCR, we kind of see a decrease, but it's not significant. And so this could also indicate why this was missed in so many other experiments. You know, will cell type specificity really matter here? Now, the next question is what happens if we take those mice who are chronically stressed and then we treat them with ketamine? And as I hope you can appreciate here, but basically once we treat mice with ketamine and we wait 2 days, we can completely reverse the changes that we're seeing in behavior as well as Kcnq2 back to baseline. So we're very surprised and happy to see this result here on a stress phenotype. These results demonstrate that this potassium channel is altered after chronic exposure, particularly in glutamatergic neurons in the ventral hippocampus, and that these effects can be reversed by ketamine. Now, it also shows that the cell type specific effects could be diluted or distorted in other brain homogeneous studies that were done up until now. Okay, so then the idea was now how are these changes happening? How is ketamine affecting the expression of Kcnq channels? And for this, we had to turn to the literature, and we found the work of Mark Shapiro at the University of Texas that very nicely show a potential mechanism that we wanted to explore. So here is a cartoon that I prepared, usually guide you through it. So it's very simplistic. On the left side, I'm simplifying 50 to 20 years of research. So it's pretty basic, but ideally we have basically ketamine binds, and then we have an influx of calcium that leads to involvement of calmodulins as calcium sensors that would lead to what we know so far about ketamine, which is involvement of mTOR, ERK, BDNF, and so on. But what we also know by the work of Mark Shapiro is that calcium sensors or calmodulin can bind in combination with ACAP5, which is a complex that leads to the activation of Kcnq channels. So the question was, can ketamine alter this mechanism? If we know that calcium influx signaling can affect transcription of Kcnq2 through this complex of ACAP5, can we see something similar? So now, something that immediately caught our attention is that if we look at the key proteins or the key genes in this pathway that I just presented here, the ones in red, we could already see that those genes were differentially expressed in our single cell data. So again, there was a good beginning. We have Kcnq2, of course, but as well as ACAP5, which is the main component of this pathway, as well as the calmodulins. The other thing that was good is that we can modulate the system in vitro. We can use inhibitors to activate or deactivate some of these proteins and see what happens through the expression of the gene. And that's what we did. It's essentially the same in vitro system that I mentioned earlier. We use primary neurons from mouse hippocampus. We treat them with ketamine, plus one of the inhibitors. We did look at three different things. We use nifedipine, which blocks the activity of calcium signaling. We use W7, which inhibits calmodulins, or cyclosporine, which basically would block the formation of this complex. And what we see is that after treatment with ketamine here in orange, light orange, we can see an increase 1 hour, 2 hours, or 6 hours later, which is essentially what we've seen earlier on. But interestingly, when we combine ketamine with the inhibitors, we basically could abolish the expression increase of Kcnq2, suggesting that there is an important role for calcium channels, calmodulin and calcineurin, and this pathway in the transcription and regulation of Kcnq2. Now, again, this result describes a previously unknown mechanism of action of ketamine through Kcnq2. Okay, then we also did some pharmacology experiments to try to push this a little bit further to try to see what happens with behavior. Again, as I said, Kcnq channels, we know of a lot of very specific inhibitors and activators. And so we tried to do is we basically took mice, treat them as we did before with ketamine, at the same time that we treat them with either an inhibitor or an activator. And we put them through a forced swim test to understand, to try to assess their behavior. Well, you see here in blue, both with inhibition and activation, the mice only receive ketamine. We see a decrease as we showed you earlier. Once we test them with two different concentration of XT991, which is a specific inhibitor of Kcnq channels, we completely abolished the antidepressant effects. But when we tried it with an activator, in this case, a medication called reticavin, which is a Kcnq activator, we see a stronger effect of the forced swim test at a lower concentration, but we lose the effect at a higher. And this is interesting, and we can discuss this later, because ketamine also has an inverse U-shape effect at lower and higher concentrations. So this, again, caught our attention. Okay, one of the things is here is we've only used so far one way to assess antidepressant treatment response, which is the forced swim test. And there's a lot of issues with this alone. So what we wanted to do is add a different way to assess treatment response. And for this, we use a novel system that we call the social box. So the social box is pretty much an enriched environment where mice live in groups, and we continuously track them, track the behavior with a focus on social interaction. So I have a video here that I will try to show you so you can sort of visualize this better. So this is an arena where our mice are colored. We can track them. We know what they're doing constantly. And we do this over time. We can do this for days, weeks, and obtain a large amount of information. So we can continually track their behaviors in multiple groups of mice, and we're able to detect behaviors that we're not able to pick up in standard paradigms. We can do this at night when the mice are active in their natural environment. So this requires very minimal experimental intervention. So it's a more or a semi-etiological environment, I would say. Stop the video not to distract you more. But basically what we do is the mice are in the arenas, we quantify their baseline behaviors for several nights, so we know how they're acting. 4 days later, we treat them with ketamine or saline, just as we did in the past in the previous experiment, and then we assess their response with the social boxes for the next 2 days. Now, we use machine learning models to try to understand what differences we can find, subtle differences in behavior. Very early on, it's really easy to see that we can separate those mice in gray here that are treated with saline versus those who in blue who are treated with ketamine. Now, when you look at this analysis, it's more like a black box. We know we're different, but how are they different? Why are they different? And that's something that we can look at what are the parameters that are making this analysis, or the components that are standing out. And these are things like the time that the animals spend away from the walls, interacting with other mice, eating in areas that are more anxiety provoking in the arena, and so on. So we can use this information to then build a predictor of how we know that mice will work or will behave after treatment of ketamine. And then the question is, okay, once we have this predictor, can we change the behavior of these mice pharmacologically by manipulating Kcnq channels? So we focus on reticavin when we see an increase or stronger effect. And here, where you can see, this is our predictor. In blue, you can see that we can separate those who are treated with ketamine versus those who are not. And as we use reticavin at a different dose, as we saw early on, at the lower dose, we can see a stronger effect than we lose at the lower effect. So again, this was very interesting, but it confirmed what we saw with classical behavioral paradigms of the forced swim test. And again, suggests that the Kcnq channels could modulate antidepressant-like effects in a semi-naturalistic living environment. So now finally, just to wrap it up, then we did a few more experiments. We were interested to know what happens. All the experiments that I mentioned are only 2 days after treatment. So what happens later? Can we still see the effects a week later? And so that's what we see. So at day 5, we can still see a very nice different, consistent with what we saw earlier. 2 days is very similar. But interestingly, at 7 days, we lose the effects with ketamine. I mean, we see a decrease, but we lose the significance here. But at 7 days, we can still see an effect with those mice who were treated with both ketamine and reticamine. We also wanted to see if we can augment the effects. And so in order to do this, we treated mice with ketamine at I didn't mention this earlier, we used 10 milligrams per kilogram, which is standard in the field for mice. But we also treat mice with lower dosages, 1 and 5 milligrams. And what we can see is that we can only see the effects at 10. Most people don't see effects at 5 or 1. However, once we treat mice with both ketamine and reticamine, we can see that at 5 days later, we can see similar effects that you would see with ketamine alone at a higher dose. And this is very interesting because we use it half of the medication. So this could have some very important application. Now, the last thing we showed here is that at 2 days later, in combination with reticamine, we could also see that the expression of Kcnq2 was higher in glutamatergic neurons of the hippocampus. This was not present in the negative fraction as we saw before. And most importantly, here in purple, these mice were treated with an SSRI with escitalopram. And we did not see any changes in Kcnq2 whatsoever with escitalopram. So these results, I mean, imply a potential synergistic effect between ketamine and reticamine. And the lack of effect on classic SSRI suggests that this might be a ketamine-specific role. Now, in terms of reticamine, we know it's a medication that has been developed for the treatment of epilepsy. But as we know from a lot of the work of Sarah Costia and James Morrow, we know that it's been shown in human depressed patients that there's an improvement in depression symptoms. We also know that, or it was reported that these effects were well tolerated and there were no serious side effects during the trials. However, something to point out, both in the work of these articles, or the work of Sarah, as well as the original papers on Kcnq3 with Alec and Friedman, they show that by manipulating or activating the Kcnq channel, you would see effects, but only after chronic treatment. And just to remind you now, in combination with ketamine, of course, ketamine is doing a lot of the fast acting, but we see stronger effects very early on. This is by acute treatment with a single injection. Now, obviously, this is quite interesting. Both medications are FDA-approved medications. So that's something that could really be tested in humans relatively fast. In fact, this is some of the after work that we're doing. Alon and I have filed a patent for the use of ketamine and reticamine, and there's some trials that will start in Israel to test this. Okay, so just to conclude, I mean, I hope I can convince you that our data suggests that the Kcnq2 gene is involved mediating the sustained antidepressant effects of ketamine, that the adjunctive treatment of ketamine and reticamine could augment antidepressant effects of ketamine, but ultimately, these results provide a deeper understanding of the complex mechanisms underlying the antidepressant effects of ketamine. And of course, I think these findings have very important clinical implications, and that's why I'm very happy to present here, because I would love to hear what your thoughts on this are. And with this, I'll just very quickly just like to thank everybody who was involved. This is, as I mentioned, this work that I did during my postdoc in Alon's lab. A lot of people involved. This was definitely not a one-man show. Lots of people and the funding agencies, of course. Lastly, I just want to let everybody know I just recently started my lab at the Karolinska Institute. A lot of the focus of my lab will be focused on stress, but also mechanisms of treatment response. So if there's anybody who, I'm looking for graduate students, postdocs, if anybody is interested, or if you know of anybody, please just point it to me, and yeah, you can follow me on Twitter, I guess. But thank you very, very much.
Thank you very much, Pablo. Great talk and very rich in data. So I just want to remind everybody, maybe somebody who joined later, that this is a new format. So I really encourage you to just ask any question you would like to ask. You can ask directly to Pablo. But in the meantime, maybe while we're still like all digesting this, such an interesting talk, I was just wondering, Pablo, if you can clarify a bit the timing. So in your experiment, was retigabine administered at the same time of the ketamine, or
Yeah,
no, go ahead.
go ahead. No, go ahead. Go ahead. Sorry.
Or you had like a time when you administered the ketamine and subsequently you administered.
Yeah, so we did, and I think this is important. It's something that we're currently testing further. We did it technically at the same time, but always injected Kcnq first. So there's a gap of a few minutes by the time they get a ketamine injection. So all the groups were injected first with will receive an injection of ketamine and then a second injection of retigabine first and then ketamine. There's something that we are testing too, because it's really interesting. What happens if they will receive a pre-treatment with Kcnq2 a week before? You know, would you prime the channels, and then will that be enough then to receive ketamine alone later on, or do you need them together? Or so the dynamics of what that is something that we're trying to tease apart. But it's a very good question.
Was there any reason why you administered the retigabine first?
We thought that perhaps we could inject them and then prime the channels slightly. We didn't know if it was going to be something. And then we did that, and we also did it at the same time, and we didn't see differences. And so I think that perhaps if we can do something earlier, that would make more sense. We also thought about what happens when you do the opposite. What if they receive ketamine first, and then later on Kcnq2? If that then later on will amplify the effects or extend them. So that's something that we're also looking in that combination of what or how we do. We play with concentrations. I think that's something that we did play, but we haven't really teased about the temporal window of how to administer it. But I would love to know what you guys thought, what you guys did with with patients. And
well, we never had, at least from my experience, ketamine plus retigabine. So I can really speak about that. But I think that, from a clinical point of view, it would really be interesting because of what you're saying, right? Ketamine has a prolonged effect, a rapid and a prolonged effect, but then eventually fades away.
Correct.
So it would really be interesting if there is something that, can extend even further.
Yeah, we also thought about, because of the first papers that I would see that would link these potassium channels to treatment. I can think of the work of Alison Friedman, of course, that new Nature Communications paper. I should mention that I said that we cannot differentiate when we treat with ketamine, with retigabine, between Kcnq and Kcnq3. I didn't show this data, but on the paper, every time that we measure Kcnq2, we also quantify Kcnq3.
Yeah.
And the effects were specific to Kcnq2. We were not seeing effects in Kcnq3. Now, I think the work of Alison Friedman at that time is not in the hippocampus. This is in the VTA, if I'm correct. So that's something to keep in mind. But also, I don't know if this matters, but I know that the way that they anesthetized the animals when they did their surgeries, they use ketamine, right? They use a higher concentration as an anesthetic. But we don't know if you're priming those channels or what happens then later on with retigabine. So every time our surgeries were not done, we don't use ketamine. We use different anesthetics to avoid that as well. But then that's it. I think of Kcnq channels here, and I don't know if you share this view. I see them as a more like an amplifier kind of thing. That, we see the effect of ketamine, but perhaps these potassium channels are amplifying the effect of ketamine. Now the question is, does it matter if we do it at that point and the signal is higher then and it lasts longer, or is it better to give it later on to push the effects, for example? I mean, these are all very interesting questions.
Sorry, and then I really want to leave the stage to also everybody else who would like to ask you a question. But so the way you're saying this is like you think it can be a synergistic mechanism. So basically, if you have both drugs, you can eventually lower the dose. Is this a bit?
Yeah, I mean, that is something that suggests in our findings, right? We use a lower dose of ketamine, and we can see the same effects. At least, I mean, obviously, take it with what it is. These are, behavior tasks that we are seeing, but we've seen it both in a classic behavior as well as a more complete set of behavioral analysis, and we see the same effect. So we can lower the concentration of ketamine and obtain the same type of effects. Now, whether we see that in humans or not is yet to be seen, but this is very promising, right? If we can lower the concentration, this could be things about side effects that would be also relevant. So this is very this is very interesting.